solve the possible.
Teacher Solomon, we can get started.
Please mute your audio. We're going to start in a minute.
The ascending need to be nothing more than and then
We have so many students in S5. Where are others?
Teacher, we can begin.
Yeah, we're going to begin.
After all the work done during the American headline investigation, has something been overlooked? Was there another option for normal purposes of rules training?
All right. Good afternoon. I hope you guys are doing well. Welcome to this class. This is Senior 5 Biology. Let me try sharing my screen and see where we can begin. This class or lesson is brought to you by Yaaka Digital Network. It is free for now, and we're conducting classes. We're conducting classes from P1 to Senior 6. So once again, you're welcome. Those who are new here, Teacher Solomon. Okay. And yeah, you're very, very, very welcome. Here are some of the offers that we have as Yaaka. You can pay a yearly fee so you can access teachers and our website and quizzes and assignments for a whole year by paying just 500,000 shillings. You can have these downloads. You can also access Educare at these particular fees. Okay, so many offers that we have, and they're very, very affordable. So please ensure to share with your parents. So who can remind us that what we looked at last week? Who remembers what? Who remembers what we looked at last week?
I do.
Yes, Faye.
We looked at eukaryotic cells, prokaryotic cells. We also did differences, similarities.
Okay, That is correct. The similarities, the differences in eukaryotic and prokaryotic cells. All right. So today we'll look at the cell theory. We'll look at the cell theory, and we shall also see the origin of cells and things of that nature. Now, so we have so many people with credit on the discovery of cells. People like Robert Hooke in 1665 initially discovered cells from tiny sliced pieces of cork. Matthias in 1838 and Theodore in 1839, they proposed the cell theory, which was later modified by Rudolph in 1858. Okay. And here is the modern ideas of the cell theory. Some of you did a great job last week on the activity where I tasked you to explain the cell theory. But here are what we know about the cell theory as of now. All non-living things are made up of one or more cells. Okay, we credit that to Schwann, whatever that name is, and Clayton from 1983 to 39, 1938 to 39. The other one is that the cell is the structure and functional unit of a living organism. We credit that to the same people, like above. The third is all cells arise from pre-existing cells by division. We credit that to Rudolph, 1858. The fourth idea or theory about cells is that cells contains hereditary information, which is passed from cell to cell during division. And all cells are basically the same in chemical composition. And lastly, all energy flow through metabolism and biochemistry of life occurs within cells. So all that combined is what we call the cell theory. It's like what we hypothesize or what we understand a cell to be or a cell to contain. Those 6 ideas. Okay. So that's the cell theory. Please remember, if you have a question, you can always ask. If you have a poor connection, you can always type it in the chat box if we can't hear you well. Now, we have exceptions or discrepancies to the cell theory. And here they are. So for example, viruses are obligate intracellular parasites capable of replicating only inside host cells using the machinery of the host. Viruses are therefore considered biotic but not organisms. Okay, that's one discrepancy, and it comes upon due to the fact of replication or being able to divide into very many others. So viruses are capable of replicating, but only when they are in the host. When they're outside the host, they cannot replicate. Therefore, they are biotic, but not organisms. Now, this discrepancy still has a discrepancy. Okay, there's some scientists who say viruses are just particles, they're not biotic. Others will say they are biotic. Okay, for example, the virus that is disturbing us right now, the coronavirus cannot replicate or cannot make more of itself while outside the host. It must be inside the host, which is a human being, for it to divide. It utilizes your cells to be able to divide. It cannot do it on itself, by itself. Okay. Cynocytic algae like Butyraria or Butyria and many fungi have a body that is a continuous mass of protoplasm with many nuclei, but without cell wall separations, i.e. are aseptate. So these ones, they are not in enclosures, okay? This type of fungus and algae, their bodies are not made up of the typical cell. It's just continuous masses of protoplasm. Okay, protoplasm is just the cell contents. These cell contents are not in an enclosure. It's just a continuous mass. Okay, they lack the cell wall separations. So that is another discrepancy. When someone comes up and says all organisms are made up of cells, well, we see these ones are made up of just a continuous mass of protoplasm and no separations. Okay, so that is another discrepancy in the cell theory. The other one is skeletal muscles have very long cells with hundreds of nuclei. Okay, so when we say, for example, a cell is made up of a nucleus and it's made up of the same type of chemical composition or similar chemical composition. Well, here are skeletal muscles that are very long and contain hundreds of nuclei. Okay, that's another discrepancy. Skeletal muscles are muscle cells, okay, mostly muscle cells, right? Also, the giant algae is an organism made up of one long cell, but with only one nucleus. That's another discrepancy right there. One cell with a very long, is a very long cell and with only one nucleus. Unicellular organisms can be considered acellular because they are larger than a typical cell, to carry out all functions of life. So this is another one where we see unicellular organisms like Paramecium, amoeba. They're capable of carrying out all these life functions, but they're made up of only one large cell or one cell, and it's typically larger than a typical cell. Okay, also that pokes holes in our cell theory a little bit. Some tissues, please mute your audio. I don't know who this is. Daniel, mute your audio. Daniel, if you can hear us, mute your audio. Okay, good. Good job. Okay, some tissues or organs contain large amounts of extracellular material, e.g. vitreous humor of the eye, mineral deposits in bone, and xylem in trees. So some cells are modified. This also pokes holes in the cell theory because our cell theory will tell us that the chemical composition is the same. Okay, but here we see that some cells are modified. When you look at the xylem in plants, you can see that it has modified cells where we have the tracheids and the vessels in the xylem. Okay, that tells you that the cell theory is not that perfect. So we poke holes on that when we say that all cells are made up of a similar chemical composition. We see some that are modified. Okay, let's look at the factors that limit cell size. Yes, if you have a question, you can always raise your hand and ask. So factors that limit cell size. Now, cell size is very important in determining the functions or determining how the cell is going to perform its functions. Okay, so on this table, we have the factor and the explanation of how each factor influences the cell size. Okay, so let's start with surface area to volume ratio. Surface area to volume ratio right here. So small cells have a very large surface area to volume ratio, while large cells have a small surface area to volume ratio. Now, a large surface area to volume ratio enables fast rate of diffusion. Okay. Someone is typing in the chat. Okay. All right. The small surface area at volume ratio will, for it to slow the rate of diffusion. Okay. Large surface area, fast rate of diffusion. Small surface area at volume ratio slows down diffusion. All right. Now, there's a question here that I can ask. Between a rat and an elephant, which one has a very large surface area to volume ratio? Who can tell us that? Between a rat and an elephant, which one has a very large surface area to volume ratio? Who knows the answer? Yes, Kelvin?
It's a rat.
A rat, very correct. Okay.
Very correct. Okay.
That's very, very correct. All right. So a rat has a larger surface area to volume ratio. This shows us that the smaller the organism is, the larger the surface area to volume ratio. And the bigger the organism is, the smaller the surface area to volume ratio. This is why we say that in a small organism, or that organism that has a large surface area to volume ratio, diffusion will be easier. Okay? Because fluids or whatever is diffusing has a very short distance to travel. Right? So that is important too. Okay. Small cells have low metabolic demands and form low amount of wastes, while large cells have a higher metabolic demands and they form a lot of, or a high amount of wastes. Okay, that is another factor or explanation of why cell size matters in organisms. So therefore, the large surface area to volume ratio in small cells enables adequate supply of oxygen and nutrients and expulsion of wastes. So these small organisms are more efficient in that type of manner. Okay. Carbon dioxide via the surface of the cell by simple diffusion, while the small surface area to volume ratio in large cells limits the diffusion, hence supply of nutrients by simple diffusion. So simple diffusion doesn't work well in organisms that are large or organisms that have a small surface area to volume ratio. Simple diffusion does not necessarily work. It's not adequate. Okay. It wouldn't meet the metabolic demands. Hence, in these animals, some large sized cells take in the substances in bulk, which we call the endocytosis, and expel substances by exocytosis in order to supplement the simple diffusion. So when you look at those large organisms, in animals especially, we'll see that the cells are capable of doing what we call endocytosis, that is the taking in of substances, and exocytosis, where we see the expulsion of waste. This coupled together with simple diffusion is perfect for those type of organisms, but not only simple diffusion. If it was only simple diffusion, it would not be capable. Other animal cells, they form finger-like projections, which we call microvilli, to increase on the surface area. Okay, just to increase on surface area, they form those tiny projections that we call the microvilli. Some cells divide when they reach a certain size to maintain suitable surface area volume ratio. So others are modified in such a way that once they reach a certain size, they just divide in order to maintain a very good surface area to volume ratio that wouldn't limit their cell functions. A note here to understand is that surface area to volume ratio particularly limits the size of bacterial cells, okay, i.e. prokaryotic cells, which are incapable of endocytosis and exocytosis. Okay. It is important for us to know that, because if these prokaryotic cells were capable of this endocytosis and exocytosis, it would be really a little bit risky for their cells from bursting and bringing in other substances that would harm them or harm their cell walls. So that is how surface area to volume ratio determines cell size. Any questions so far? I'll pause a little bit so that you can ask questions. Any questions about surface area to volume ratio? Remember, feel free to type them in the chat box, and I can always look at them, or you can just raise your hand and ask. Let's look at another factor. Pretty sure it won't be as big as surface area to volume ratio. Let's look at nucleocytoplasmic ratio, and we'll understand what that means. Nucleocytoplasmic ratio. Now, in a cell, we see that there is DNA, which is found in the nucleus, and it provides the instructions for protein synthesis, hence controls activities of the cell. Each nucleus can only control a certain volume of cytoplasm. Okay, so here we see that the nucleocytoplasmic ratio, it is like a gauge, right? A certain nuclear size or nuclear capability will be able to only control a certain amount of cytoplasm. Okay, specialization forms some long or large cells. Therefore, when you look at some cells that are specialized, they are specialized to overcome this limitation. Such cells are modified to become multinucleate or synocyte. E.g., the skeletal muscles that we looked at prior and fungal hyphae, they have multiple nuclei to be able to control a large amount of cytoplasm, right? Remember skeletal muscles. Why would skeletal muscles need a lot of cytoplasm? Who can tell me the answer to that? Why would skeletal muscles need a lot of cytoplasm? Besides Kelvin, who can tell me why skeletal muscles will need a lot of cytoplasm? I want someone who has not said something yet. Is there anyone who can tell us why skeletal muscles would need a lot of cytoplasm? Okay, Kelvin, tell us.
I think because skeletal muscles perform a lot of work, like activity in moving like the bones and something, so they require a lot of energy. So the existence of the much cytoplasm, it means that it will create a larger surface area for metabolic reactions which produce energy, which support the work being done by the skeletal muscle.
100% correct. Well done, Kelvin. Yes, that is correct. Skeletal muscles need a lot of energy to do their work. As we know, they are very, very, very vital in your movement and very many other exercises. So that requires a lot of energy. Therefore, they are going to need a lot of cytoplasm to house the mitochondria and other organelles that are involved in formation of energy. Okay, and this is why you'll find out that they have multiple nuclei to facilitate that. Okay, good job, Kelvin, that was correct. So this is how the nucleocytoplasmic ratio affects the cell size. The other factor is fragility of the cell membrane. As cell size increases, the risk of damage to the cell membrane also increases. This limits the maximum size of cells, especially animal cells which lack cell walls. So especially this is mainly in animal cells where we don't see a cell wall. The bigger the cell, the riskier it gets for the cytoplasm to be penetrated into through the cell membrane. Okay, so therefore some cells have actually evolved or been modified in such a way that when they reach a certain size, they just divide, they form more cells. Okay, just to avoid that risk of damage to the cell membrane. But others cannot, therefore they stop growing at a certain size. So what we call fragility of cell membrane. Someone left their audio on, it is disturbing a little bit. Technopop 2+. Okay, good. All right. The other factor is mechanical structures that hold the cell together. Cells with tough cell walls, for example, blood cells, are larger than cells with only the fragile cell membrane. Animal cells, because the tough cell walls provide support and maintain cell shape. When you look at plant cells, they are larger, they have a very large structure. So this enables them to grow at, larger sizes compared to animal cells because they already have a cell wall to protect them. They don't have to worry about protection. Other cells with complex internal cytoskeleton are larger than cells with little cytoskeleton because the cytoskeleton protects and supports the cell structure and maintains its shape. So cytoskeleton also plays a role in determining cell size. Those that have a lot of cytoskeleton are well protected. Okay, now let's look at something else. Origin of eukaryotic cells. Let's look at the endosymbiotic theory. So this one was proposed by Lin in 1967. The endosymbiotic theory suggests that the mitochondria and the chloroplasts were once separated existing in small aerobic bacteria and photosynthetic bacteria, respectively. So these were living on their own. Okay, one of the reasons we see that is because they all contain small DNA particles, circular DNA, and we shall see that later. Larger anaerobic bacteria engulfed the smaller bacteria by the process of endocytosis, but digestion failed. So here this theory tells us that the larger anaerobic bacteria engulfed the smaller bacteria by the process of endocytosis, but there was no digestion that occurred. So the smaller bacteria and the larger bacteria just created a relationship, and they remained inside one another. Initially, the smaller bacteria could have lived inside the larger bacteria either as parasites or phagocytotic vesicles, after which mutually benefiting relationship called endosymbiosis resulted, where the larger cell provided protection and shelter while the smaller organism removed oxygen, which was toxic to the anaerobic larger cell. So they developed that endosymbiotic relationship where the smaller one was able to get protection and also absorbed the oxygen that was being toxic to the larger anaerobic bacteria. Remember, anaerobic bacteria won't need oxygen for metabolism. Oxygen will actually be toxic to it. So the smaller one was able to remove that oxygen, hence that symbiotic and symbiotic relationship. This is even the reason why we call it the endosymbiotic theory. Okay. Well, something was funny. Okay. Something is happening here. This is too far. Okay, we need to get back the other side. All right. Before we look at all this, yes, right here. Sorry about that. So with time, the mitochondria and chloroplasts were modified into organelles suited for respiration and photosynthesis inside the larger eukaryotic cells. Okay, this was the result. Note secondary endosymbiosis involves a larger eukaryotic cell engulfing a smaller eukaryotic cell. There's something to note about the endosymbiotic theory. Sometimes it does not involve the anaerobic one engulfing the aerobic one. Sometimes just between a larger eukaryotic cell engulfing a smaller eukaryotic cell. Okay. So here is an illustration of the endosymbiotic theory. Let's see. So here we see the unfoldings in the plasma membrane of the ancestral cell give rise to the endomembrane components, including the nucleus and endoplasmic reticulum. Okay. So the original cell or ancestral cell evolved was through those foldings inside its cytoplasm, which resulted into the development of the nucleus right here and the endoplasmic reticulum. Someone has written something. Okay, seeing the message. All right. So this is what happened in the original cell. But also in a first endosymbiotic event, the ancestral eukaryote consumed the aerobic bacteria that evolved into the mitochondria. So this is what happened here. Okay, the aerobic bacteria was taken into the ancestral cell by endocytosis. This bacterial cell developed into the mitochondria. In the second endosymbiotic event, the early eukaryote consumed photosynthetic bacteria that evolved into the chloroplast. So here we see the photosynthetic bacteria being swallowed or taken in by the early eukaryote cell. Now this developed into the chloroplast that we see today, according to this theory. Then after that, we see the modern photosynthetic eukaryote, okay, which contains the chloroplast. Now the one that didn't swallow the photosynthetic bacteria developed into the photosynthetic eukaryote. For it developed into a modern heterotrophic eukaryote. This one wasn't able to swallow or to take in a photosynthetic bacterium. So for it remained with only the aerobic bacterium that evolved into the mitochondria. So this one evolved into the heterotrophic eukaryotic cell we see today. So that is the illustration of the endosymbiotic theory. Okay. Any questions about that? Yes, Kelvin.
Does it mean that the photosynthetic eukaryote is the plant cell and the other one is the animal cell?
Exactly, that is correct. The photosynthetic eukaryote went on to make the plant cells, okay, to become plant cells and develop into the plants. And then the modern heterotrophic eukaryote evolved into the animal cells to form the different animals we see today that have that type of eukaryotic cell. So yes, the answer is yes to that. Good question, Kelvin. That's a good question. Okay. Any other question before we continue? So here are the evidences. Okay. The evidences for this endosymbiotic theory. We see that the mitochondria and chloroplasts have their own DNA. I mentioned this at the start. And they divide independently from the cell they live. So they have their own independent DNA. It's circular DNA, which is found in these two organelles. We see that also in today's bacteria. Most of them have circular DNA, okay? And it's not enclosed in the nucleus. That is a pattern that we see also in the mitochondria and the chloroplast. So that is one evidence. And there is a great similarity between prokaryotic cells and organelles of the eukaryotic cells. This is exactly what I'm mentioning, that there is a very good similarity between today's prokaryotic cells and the organelles that are found in eukaryotic cells. So this gives us some sort of confidence in the theory of endosymbiosis. Okay, so this more evidence here gives us some confidence in that theory. All right. So let's look at these features between prokaryotes, eukaryotes, mitochondria, and chloroplasts of photosynthetic eukaryotes. These are some of the similarities and differences that we can see between all those four different cells and organelles. The two are organelles, the two are cells. Let's look at their DNA. So the DNA of prokaryotes, is one circular chromosome. Okay, so this is circular DNA in here. in prokaryotes. And then in eukaryotes, they are linear chromosomes. Okay, many chromosomes connected together and they're linear. But in mitochondria, we see circular DNA and in chloroplast. So this gives us the feeling that these are similar to the prokaryotes, right? And the replication here we see in prokaryotes is binary fission. One cell splits into two, but in eukaryotes we see mitosis, right? In the mitochondria and the chloroplast, similarly to the prokaryotes, they also do binary fission. Okay, one splits into two. So these organelles divide independently, and that's how they divide. Their division is similar to the prokaryotes, as you can see. That's a good evidence also supporting the endosymbiotic theory. The ribosomes, we have the 70S in the prokaryotes, 80S in the eukaryotes. But look here, in the mitochondria and the chloroplast, we have the 70S, similar to the prokaryotes. Okay. When we look at the electron transport chain, if you have not studied this before, you'll study it very soon in respiration or even in photosynthesis. So the electron transport chain occurs in the plasma membrane for the prokaryotes. It occurs in the plasma membrane. But in eukaryotes, we see it in the mitochondria and chloroplast, not in its plasma membrane, okay, in the eukaryotic cells. It doesn't occur in its plasma membrane, but in the membrane of mitochondria and chloroplast. So in the mitochondria, it occurs in the process of respiration, and in the chloroplast, it occurs in the process of photosynthesis, which is the same here in the individual organelles, right? So we see that similarity with also the prokaryotes. The sizes, approximately 1 micrometer to 10 micrometers. This one is 50 to 500, but the sizes of mitochondria and chloroplast are similar to that of the prokaryotes. So again, we see that pattern of similarity in prokaryotes and organelles of eukaryotic cells. Okay. The other evidence here we see is that the timeline of life on Earth shows that from fossil evidence of bacterial life, the mitochondria, chloroplast, and eukaryotic cells emerged about the same time, a few billion years ago, right? So about 1.5 billion years ago, we see the emergence of the mitochondria, chloroplast, and eukaryotic cells. So it's about the same time. This shows us that this is also another strong evidence. The prokaryotes appearance on Earth was about 3.8 billion years ago through the photosynthetic bacteria, 3.2 billion years ago through the anaerobic bacteria, okay, and 2.5 billion years ago for the aerobic bacteria. But for the eukaryotic cells, we see it is about 1.5 billion years ago. And for the mitochondria, similar, about 1.5. Meaning that the eukaryotic cells and their organelles emerged around the same time. Okay, so probably the endosymbiosis theory is true due to endocytosis of these mitochondria and chloroplast into the eukaryotic cells. So those are the evidences, okay? It's the same thing that is in the table right there. Let's look at the semiautonomous organelles. We see the mitochondria DNA and the chloroplast DNA. The shorthand provides more part in the genome needed for binary fission. So these organelles are controlled by a nucleus, which contains a larger genome. So the control of the entire genome of the cell is by the nucleus, but even though the mitochondria and the chloroplast have their own DNA, that just helps in binary fusion. Mitochondrial DNA and chloroplast DNA is short, therefore can only code for a few of the proteins needed. Hence, some of the required proteins are imported from the cytoplasm of the main cell where the organelle is. So these are like some of the shortcomings. And last but not least, this is the generalized structure of cells as observed under a light microscope. Next time we shall look at the bigger structures that are seen in an electron microscope, where we have other organelles. But this is the simple animal cell as viewed under a light microscope. We have the cell membrane, cell vacuoles, the nucleus, and the cytoplasm. These are only seen by stains. Okay, we stain, and then we can see these few features. For the plant cell, this is what we are able to see. We see the cell wall, the cell membrane, cytoplasm, nucleus, vacuole, and chloroplast. And all this is under a light microscope. So the next time we meet, we shall look at the comprehensive structures. Okay. What is a genome? That's a very good question. Who can answer that for us? What is a genome? What is a genome? Who knows the answer to that? Someone is asking in the chat box what a genome is. So a genome is just information, a collection of information. We normally find this information in DNA. This information is carried on from one cell to another. So these are like genes, right? The information is in form of genes in DNA that is carried on from one cell to another through cell division. That is a genome, okay? So this information, a genome can be a genome of a population, right? The information that is for a specific type of species, right? So it will depend on that. You're welcome, Kelvin. Any other questions before we go? Questions? Okay, I'm going to send out these notes for you to go through, so don't worry about it. And if you have questions, always feel free to ask in the group, or you can inbox me and I'll answer you. Well done. Good job, everyone. So let's come back here for chemistry very soon. In chemistry, we shall look at mole concept, or the mole. So be prepared for that to see some calculations. So come back here at exactly 1 p.m. for chemistry. See you soon.